Hey champs of Examsnet, welcome to the question series of Examsnet. I hope this series of detailed explanation is helping you a lot. So today we are going to discuss NEET 2016 biology paper of phase 2. So let's start the session. The first question is from the chapter Biotechnology Principles and Processes. And the question is, a foreign DNA and plasmid cut by same restriction endonuclease can be joined to form a recombinant plasmid using options are ligase, EcoR1, Taq polymerase and polymerase 2. And the correct answer is option A, that is ligase. Okay. So we know that ligase is also known as molecular glue. Molecular glue. So it acts as a glue or pasting agent. And what does it do? It is used to join the substrates. Okay. Ligase is an enzyme that joins the substrates. And in this case, DNA, that is here the DNA has to be joined, right? So here T4 DNA ligase is to be used, okay, for joining the ends of DNA fragments. So option A will be the correct answer. Question number 2 is again from the chapter Biotechnology Principles and Processes. And the question is, which of the following is not a component of downstream processing? Options are expression, separation, purification and preservation. So what is meant by downstream processing? See, after the completion of the biosynthesis of a product, the product has to be subjected through series of processes before it is ready for marketing as finished good, right? So first what is done? Biosynthesis of the product is done. Biosynthesis of the product. That means preparation of the product. And then there are certain processes, series of processes before the marketing. Okay. And then it is ready as finished good. And then that finished good is sent to the market. Okay. So it is sent for marketing. Now, these series of processes are known as downstream processes. Okay, downstream processes. And what are the components of downstream processes? These are basically separation and purification. Okay, mainly separation and purification are the components of downstream processes. But after that, preservation also has to be done. Okay, they have to be preserved using suitable preservatives, and then quality control testing is also done. So here in the given options, three things are mentioned: separation, purification and preservation. So these are the components of downstream processing, but the expression is not the component of downstream processing. Here expression is about the expression of recombinant DNA or expression of protein, that is part of upstream processing. Okay, not downstream processing. Upstream processing, it includes the growing of the biomolecules. Okay, so option A will be the correct answer. Now question number 3 is again from the same chapter, biotechnology, and the question is, which of the following restriction enzymes produces blunt ends? Options are hint 3, SAL1, EcoRV and XHO1. So what are blunt ends? See, when DNA is cut using restriction enzymes, then two types of ends can be created. First type is sticky type. Okay, sticky type of end or sticky end. And the second type is blunt end. Now, what is the difference between these two? In case of sticky ends, one strand is longer than the other. Okay, suppose the upper strand is longer, then the lower strand will be smaller. Okay, and this side, the other DNA will have the upper strand shorter and the lower strand longer, so that these two can stick together. Okay, whereas in case of blunt end, both the ends are of equal length. So what can we say? That in case of sticky ends, the upper strand has more number of nucleotides as compared to the lower strand, whereas in case of blunt end, both the strands are having equal number of nucleotides. Okay, therefore these are of equal length, right? Now here if we talk about the options, except eco RV, all are having sticky ends. Okay, Hind 3, Sal 1 and XHO1, they have sticky ends. Only eco RV produces blunt ends. Okay, so option C will be the correct answer. Question number 4 is from the chapter Biotechnology and its applications, and the question is, which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase, that is ADA deficiency? Options are radiation therapy, gene therapy, chemotherapy and immunotherapy. So the correct answer is option number B, that is gene therapy. When gene therapy is used, it is used in case of hereditary diseases. Okay, when the disease is hereditary, then we use gene therapy. Okay, gene therapy. So it is basically used for the correction of gene defects. Correction of gene defects. So this is the therapy that the question is asked about. Okay, so in 1990, a 4-year-old girl with ADA deficiency was treated with the therapy known as gene therapy. Okay, if we talk about other options, that is radiation therapy, chemotherapy and immunotherapy, these are types of therapies which are used in case of cancer treatment. Okay, in case of radiation therapy, high doses of radiations are used to kill cancer. In case of chemotherapy, chemicals are used. Chemicals are used for cancer treatment. And in case of immunotherapy, what is done? It is also used for cancer treatment. And here white blood cells are used. Okay. So the correct answer is option number B, gene therapy. Question number 5 is from the chapter Biodiversity and Conservation, and the question is, how many hotspots of biodiversity in the world have been identified till date by Norman Myers? Options are 43, 17, 25 and 34. So the correct answer is option D, that is 34. Okay. What is biodiversity hotspot? See, for an area to be identified as biodiversity hotspot, it has to fulfill 3 criteria. Okay. So the region or the area should have 3 characteristics. First one is high biodiversity. High biodiversity. That means there is a large biodiversity in that area. Okay. Then the second characteristic is high degree of endemism. High degree of endemism. That means endemic species are present there, and endemic species are those species which are not found in any other area. Okay, they are found only in that particular area, right? And then the third one is threat of extinction. Okay, threat of extinction. So these species are highly vulnerable to extinction. Okay, so if the region has these 3 characteristics, it can be identified as hotspot of biodiversity or biodiversity hotspot. Okay. So here we can see that there are total 34 biodiversity hotspots in the world that have been identified by Norman Myers. Okay, out of these 34, 3 are present in India. Okay, 3 are in India and total 34 in the world. So option D will be the correct answer. Now question number 6 is from the chapter ecosystem, and the question is, the primary producers of the deep sea hydrothermal vent ecosystem are, options are coral reef, green algae, chemosynthetic bacteria and blue-green algae. Okay, so let's first understand what is deep sea hydrothermal vent. So as the name suggests, deep sea means it is located at great depth in the sea or in the ocean. Okay, and vent basically means an opening or a crack. Okay, crack or opening. So we can say that these are cracks in the ocean. Okay, cracks in deep oceans. Now hydrothermal. So hydro is related to water and thermal is related to heat, right? So what happens at these locations, hot water is coming out of the cracks, okay? Hot water is coming out. So due to that, the temperature is very high here, okay? So high temperature is there. So this is about the deep sea hydrothermal vent. Now, what kind of organisms can thrive in such extreme condition? So we know that special type of bacteria known as archaeobacteria can thrive in extreme conditions, okay? Extreme conditions, because they have specialized structures for their protection, okay? So here, out of the given options, the chemosynthetic bacteria will be the correct answer, because these are the examples of archaeobacteria, okay? Chemosynthetic bacteria. So here the question is asked about the primary producers. So these chemosynthetic bacteria also act as the primary producers in these locations, because these are autotrophic in nature. Okay, these are autotrophs. They can synthesize their own food. But as we know that in such a great depth, at such a great depth, sunlight will not be available. Okay, so how will they synthesize their own food? They will use the chemical reactions. Okay, chemical reactions. Therefore, these are known as chemosynthetic bacteria, because they use inorganic chemical reactions. So instead of oxygen, the product will contain sulfur compounds. The products are mostly sulfur compounds. Okay. For example, hydrogen sulfide. So the correct answer is option number C, chemosynthetic bacteria. They act as primary producers of the deep sea hydrothermal vent ecosystem. Question number 7 is from the chapter Organisms and Population, and the question is, which of the following is correct for R selected species? So what are R selected species? See, R is related to reproduction, okay, or it is related to more reproductively capable species, okay. So we can say that R selected species have high reproductive capacity, okay, high reproductive capacity. That means with minimum effort and in very less time they can produce a large number of progeny, okay. So minimum effort is required, their gestation period is very less, okay. So they require very less time, okay, less time is required, and they produce large number of progenies, and such progenies are very small in size, okay. So they are small in size. So the correct option will be option number B, that our selected species have large number of progeny with small size, okay. Question number 8 is again from the chapter Organisms and Population, and the question is, if plus sign is assigned to beneficial interaction, minus sign to detrimental, and zero sign to neutral interaction, then the population interaction represented by plus minus refers to. Options are parasitism, mutualism, amensalism, and commensalism. So we know that plus sign means the organism is benefited and minus sign means the organism is harmed. Okay, so plus minus means one organism is benefited and the other is harmed. Okay, so we can say that in this kind of interaction, one organism or one species is benefited at the expense of another. Okay, at the expense of another. That means the other species will either die or it will face a loss or harm of some kind. Okay, and the correct answer for this kind of interaction will be parasitism. Okay, this involves the benefit of one organism and the loss of another organism. Okay, if we talk about the other options, the second one is mutualism. So it is an interaction where both the organisms are benefited. Okay, these both are beneficial for each other, right? So this is a plus plus kind of interaction. Then the third one is amensalism. Here one is benefited and the other is neither benefited nor harmed. So it is neutral. It is unaffected. Okay. So it is plus and zero kind of interaction. Whereas in case of commensalism, one is harmed and the other remains unaffected. So this is minus and zero kind of interaction. Okay. So here it was asked about plus minus. So the correct answer is parasitism, option number A. Question number 9 is from the chapter Biodiversity and Conservation, and the question is which of the following is correctly matched? In the first option, stratification is matched with population. So what is stratification? It is the vertical distribution of habitat or vegetation. Okay, so we can say that this is a structural component of ecosystem. Structural component of an ecosystem, where the vegetation or the habitat is distributed vertically. Okay, so this should not be matched with population, so this is not a correct match. Now in the second one, parenchyma is matched with opuntia. Now what is parenchyma? It is a type of parenchyma tissue. Okay, let's write it down here. Parenchyma, so it is a type of parenchyma tissue, parenchyma, having fluid filled, air filled cavities. Okay, air filled cavities. Now what do these air filled cavities do? They provide buoyancy to the plant. So if the plant is in water, it is an aquatic plant, then it will provide buoyancy to this plant and prevent it from sinking in under the water. Okay, so it provides buoyancy to the plant. So we can say that this is a characteristic of an aquatic plant, or more specifically hydrophytes. Okay, hydrophytes. Okay, and here it is matched with opuntia, which is not a hydrophyte, instead it is a xerophyte. Xerophyte are those plants which grow in dry condition, not in aquatic condition. So this is also not a correct match. Now in the third one, age pyramid is matched with biome. So age pyramid is type of representation of age of a population in the form of pyramid. So this is related to population, not biome. Okay, so this is also not a correct match. Now in the last one, Parthenium hysterophorus is matched with threat to biodiversity. Now this is a correct match. Parthenium hysterophorus, it is also known as, let's write it down here, as the carrot grass. Okay, carrot grass or the congress grass. So basically it is a type of invasive weed species. Okay, invasive weed. So it grows so fast that it does not allow the growth of any other plants near it. Okay, so this is a threat to biodiversity. Therefore option D will be the correct answer. Question number 10 is again from the chapter Biodiversity and Conservation, and the question is red list contains data or information on. Options are marine vertebrates only, all economically important plants, plants whose products are in international trade, and the last one is threatened species. Okay, so the correct answer is option D, that is threatened species. This is a direct question. We know that red list is a type of book that contains data on threatened species of plants and animals. Okay, so option D will be the correct answer. Question number 11 is from the chapter Biological Classification, and the question is which one of the following is wrong for fungi? So we have to find the wrong statement. Okay, the first statement says they are both unicellular and multicellular. So this is a correct statement because fungi can be unicellular as well as multicellular. Okay, then the second statement says they are eukaryotic. This is also a correct statement because fungi have a well-defined nucleus. Okay, well-defined nucleus along with a nuclear membrane, and within this nucleus the hereditary material or the DNA is present. Okay, then the third statement says all fungi possess a purely cellulosic cell wall. This is not true. Some fungi possess a cellulosic cell wall, but not all. Generally, the cell wall of fungi are composed of chitin, okay, not cellulose. But there are some exceptions. For example, oomycetes, which belong to phycomycetes. So these have cellulosic wall. Okay, these have cellulosic cell wall. But that doesn't mean that all fungi possess a cellulosic cell wall. So this is not correct. Okay. Then the last statement says they are heterotrophic. This is correct. They cannot synthesize their own food. They have to derive it from outside. Okay. So the only incorrect statement is the third one. So option C is the correct answer. Question number 12 is again from the chapter biological classification, and the question is methanogens belong to, options are slime molds, eubacteria, archaebacteria and dinoflagellates. So methanogens are a type of bacteria, right? So they belong to which phylum? Monera. Monera phylum contains bacteria. Now these bacteria can either be archaebacteria or eubacteria, okay? Archaebacteria or eubacteria. Archaebacteria are ancient form of bacteria which are able to thrive in extreme environmental conditions. Okay, extreme environmental conditions, because they have specialized structures in order to survive in these conditions. Whereas eubacteria are more modern form of bacteria, and these are also known as true bacteria. Okay, true bacteria. But here the question is asked about the methanogens, so they belong to the ancient form of bacteria, that is archaebacteria. So the correct answer will be option C, that is archaebacteria. Okay, and methanogens are known for producing biogas. Biogas, and biogas contains methane, therefore these are known as methanogens, and these are obligate and aerobe. And aerobe, that means they do not require oxygen for respiration. Okay, now, if we discuss other options, slime molds belong to which phylum? They belong to Protista, not Monera. Eubacteria we have already seen. These are also type of bacteria and belong to Monera phylum. Dinoflagellates also belong to Protista phylum. So, according to the question, the correct answer is option C, Archaebacteria. Question number 13 is again from the chapter biological classification, and the question is select the wrong statement. So here also we have to find the incorrect statement. The first statement says diatoms are microscopic and float passively in water. So this is a correct statement. So let's see what are diatoms first. Diatoms are unicellular and eukaryotic organisms. Okay, unicellular and eukaryotic. Therefore, these are very small. So these are microscopic. Okay. And these can be marine as well as freshwater. So these are marine as well as freshwater organisms. And these belong to which kingdom? They belong to the kingdom Protista. Now, the statement says that these are microscopic. This is true. And they float passively in water. This is also true, because these are a kind of phytoplanktons. Okay. Phytoplanktons. Therefore, they float along with the water current and they do not require energy for floating. Therefore, these are they float passively. Okay. Now, the second statement says the walls of diatoms are easily destructible. This is absolutely wrong, because the walls of diatoms are made up of silica. Okay. Walls are made up of silica along with the cellulose, and this is non-destructible. Non-destructible in nature. So what happens when they die? It gets deposited under the water. Okay, so it is deposited at the bottom of the water body. Okay, and it forms diatomaceous earth. Diatomaceous earth, because it contains the cell wall of diatoms. Okay. Now let's come to the third statement. It says diatomaceous earth is formed by the cell walls of diatoms. Yes, this is true. We already saw this. Then the last statement says diatoms are chief producers in the ocean. This is also correct. Okay, because these are phytoplanktons. So these are producers, and these are the chief producers in the oceans. So we can see that all are correct except the second one. So option B is the correct answer because this is the incorrect one. Question number 14 is from the chapter The Living World, and the question is the label of herbarium sheet does not carry information on. Okay, does not carry. So the options are height of the plant, date of collection, name of collector, and local names. So the correct answer is height of the plant because other things are mentioned in the herbarium sheet. And what is a herbarium sheet? It is a simple sheet of paper like this and a plant sample. Okay, some part of the plants are pasted here which have been dried and pressed for a long time, and then some informations about this plant species has been mentioned on the herbarium sheet. Now these informations can include the date and place of collection, date and place of collection, then the names of the plant in English, local names and botanical names. Okay, botanical names of that plant species, then family of the plant, the family to which it belongs, the name of the collector, okay, the person who has collected that sample, okay, etc. But it does not carry information on the height of the plant. So option A will be the correct answer. Question number 15 is from the chapter Plant Kingdom, and the question is conifers are adapted to tolerate extreme environmental conditions because of options are presence of vessels, broad hardy leaves, superficial stomata and thick cuticle. So what are conifers? These are gymnosperms, okay? Gymnosperms, and these are xerophytic plants. Xerophytic. That means these are found in the dry conditions. So there is a lack of water. So they have to survive in that extreme environmental condition or extreme dry condition. So in order to survive there, they have certain adaptive features. For example, they have thick cuticles, thick cuticles. Then they have sunken stomata. Then they have needle-like leaves. So how do they help? See, needle-like leaves, that means they don't have a broad leaf, and through leaf the water is evaporated. So when the leaf is very thin, needle-like, then there is less evaporation of water. So that is used to prevent the water loss. Okay. Similarly, sunken stomata prevents the water loss from the transpiration. Okay. So this way they are able to prevent water loss and preserve water as much as they can. So the correct answer will be option D, that is thick cuticle. Because of this, they are able to adapt in the extreme environmental conditions. Question number 16 is from the chapter Plant Kingdom, and the question is which one of the following statements is wrong? First statement says laminaria and sargassum are used as food. This is a correct statement. Laminaria and sargassum are types of brown algae, okay? Brown algae or phaeophyceae, and these are used as food. Then the second statement says algae increase the level of dissolved oxygen in the immediate environment. This is also correct because algae are photosynthetic organisms, okay? So they perform photosynthesis, and due to that they release oxygen in the immediate environment, okay? Third statement says algin is obtained from red algae, carrageen from brown algae. This is incorrect. Both parts of this statement are incorrect because algin is not obtained from red algae. Algin is obtained from laminaria and fucus, okay? Laminaria and fucus, and both are examples of brown algae, not red algae. Then the second statement, the second part of this statement is that carrageen is obtained from brown algae. This is also incorrect because carrageen is obtained from a red algae, not brown algae. Okay, red algae, and that is known as Chondrus crispus. Chondrus crispus. Okay. The last statement says agar agar is obtained from Gelidium and Gracilaria. This is also a correct statement. Agar-agar is a gel-like substance and it is obtained from Gelidium and Gracilaria, which are type of red algae or rhodophyceae. Okay. So the only incorrect statement is the third one, that is option C. So this is the correct answer. Question number 17 is from the chapter Morphology of Flowering Plants, and the question is the term polyadelphous is related to, options are calyx, gynoecium, androecium and corolla. So the term polyadelphous is made up of two words, poly and adelphous. Okay. Now poly means many and adelphous is used for fused stamens. Fused stamens. So when the stamens of a flower are fused through the filament, this condition is known as polyadelphous. Okay. And we know that stamen is part of the male reproductive part of the plant, right? Male reproductive part, that is known as androecium. And gynoecium is the female reproductive part. So here the question is polyadelphous, that is related to the male reproductive part, androecium. So the correct answer will be option number C, androecium. Question number 18 is again from the chapter Morphology of Flowering Plants, and the question is how many plants among Indigofera, Sesbania, Salvia, Allium, Aloe, Mustard, Groundnut, Radish, Gram and Turnip have stamens with different lengths in their flowers? Options are 6, 3, 4 and 5. So we have to identify the plants which have stamens with different lengths. Okay, so different lengths of stamens, different lengths of stamens. This characteristic is found in two families. First one is Laminaceae and the second one is Brassicaceae. Okay. Now let's see out of the given options which ones belong to these two families. The first one is Indigofera. It belongs to Fabaceae family. Sesbania also belongs to Fabaceae family. Then we have Salvia. It belongs to Laminaceae family. So one is added here, Salvia. Okay, it belongs to Laminaceae. Then we have Allium and Aloe. These two belong to Liliaceae family. Then the next one is Mustard. So mustard belongs to Brassicaceae family. So one added to Brassicaceae, that is mustard. Okay. Then the next one is groundnut. Groundnut also belongs to Fabaceae family. Then the next is radish. Radish belongs to Brassicaceae. So one more added in Brassicaceae here, that is radish. Next one is gram. It also belongs to Fabaceae family. Then we have turnip. Turnip also belongs to Brassicaceae. So one more added here, turnip. Okay. So out of all these, we have 4 plants. One is salvia and 3 mustard, radish and turnip. So these 4 have different lengths of stamens because salvia belongs to Laminaceae and mustard, radish and turnip belong to Brassicaceae. So total 4 plants. Okay. So the correct answer will be option number C, 4. Question number 19 is also from Morphology of Flowering Plants, and the question is radial symmetry is found in the flowers of, options are Cassia, Brassica, Trifolium and Pisum. So the correct answer is option B, that is Brassica. So okay, so Brassica has radial symmetry and this condition is known as actinomorphic. Okay, so this is an actinomorphic flower, whereas all other 3 are zygomorphic in nature. Okay, zygomorphic. That means these have bilateral symmetry. Bilateral. These also have zygomorphic flowers. Actinomorphic means radial symmetry. Now what is the difference? In case of zygomorphic, you will get 2 equal halves only in one plane. Okay, whereas in case of radial symmetry, 2 equal halves are obtained, 2 equal halves in more than one plane. In more than one plane, because Here the symmetry is about the central axis. Okay, so the correct answer according to this question is option number B. Question number 20 is again from the same chapter, Morphology of Flowering Plants, and the question is free central placentation is found in, options are citrus, dianthus, argemone and brassica. So what is placentation? It is the arrangement of ovules inside the ovary. Okay, placentation means arrangement of ovules inside the ovary. Now there are several types of placentation. First one is the marginal placentation. Okay, marginal. So as the name suggests, here the ovules are arranged or inclined on the margin of the ovary. Okay, like this. If this is the ovary, then ovules are inclined on the margin. And the example of such kind of placentation is P. Then the second type of placentation is axial placentation. And here also you can understand by looking at the name axial, looking at this word axial, because in this kind of placentation there is an axial placenta, that means on the axis. Okay, so axial placenta is there like this here. Okay, and the ovules are arranged in the multilocular ovary. Okay, so the example of this kind of placenta is citrus. Okay, now let's move to the third type of placentation, and that is parietal. Okay, parietal placentation. So what happens in parietal placentation? Here the inner wall of the ovary has ovules at the periphery. Okay, so ovules are on the periphery of the ovary. And the example of this kind of placentation is argemone and brassica. Both are mentioned in the options here, right? Fourth type is free central placentation. Free central placentation. And here, as the name suggests, the ovule is located at the center of the ovary. Okay, center of ovary. And the example is dianthus. The last one, the last type of placentation is basal placentation. Okay, so basal, that means at the base of the ovary. The ovules are located at the base of the ovary. Now here the question is asked about the free central. So we have seen that the example of free central is dianthus. So the correct option will be option number B, dianthus. Okay, citrus we have already seen, it is the example of axial placentation. And argemone and brassica are they have free parietal placentation. Okay, parietal. Question number 21 is from the chapter Anatomy of Flowering Plants, and the question is cortex is the region found between, options are endodermis and vascular bundle, epidermis and stele, pericycle and endodermis and pith. Okay, so let's see. If we look at the transverse section of a stem, then the layers of cells are arranged in the following manner. The outermost layer consists of epidermis. Outermost layer of cells is epidermis. Okay, then the next one is hypodermis. Again, if we move inside, we'll get cortex. On further moving inside, we'll get endodermis. Then inside the endodermis, we have stele. Now stele contain pericycle, then vascular tissues, and then pith. Okay, so these three are together known as stele. Now let's see. The question is asked about the cortex. Okay, so the first option is endodermis and vascular bundle. That means this and this. So obviously cortex is not present between these two. Okay, so this is not the correct answer. Second one is epidermis and stele. So yes, one side there is epidermis and on the other side we have stele. So this can be correct answer. Third one is pericycle and endodermis. Again, pericycle is here and endodermis is here. So it is saying that it is present between these two. Again, this is not possible, okay, because endodermis and pericycle both come after cortex. Then the next one, the last one is endodermis and pith. Again, these two come after cortex, okay. So cortex is present between epidermis and stele. So option B will be the correct answer. Question number 22 is again from Anatomy of Flowering Plants, and the question is, the balloon-shaped structures called tyloses are linked to the ascent of sap through xylem vessels, originate in the lumen of vessels, characterize the sapwood, and the last one is, are extensions of xylem parenchyma cells into the vessels. Okay, so the correct answer is option number D, that is, the tyloses are extensions of xylem parenchyma cells into the vessels. Okay, so this is actually the definition of tyloses. Tyloses are nothing but the ingrowth or the extension of parenchyma cells into the xylem vessels. Okay, xylem vessels. So due to that, these become non-functional. That means these are not able to transport water and minerals. Okay, water and minerals are not able to be transported into the xylem vessels. Okay, so these are non-functional, and also due to the deposition of some pigments, such as tannins, resins, etc., they become dark in color. Okay, and this is a characteristic of heartwood. So let's see, the first option says that they are linked to ascent of sap through xylem vessel. This is not true. Okay. Second one says originate in the lumen of vessel. No, they originate in the parenchyma cells. Okay. Third one says characterize the sapwood. No, we have seen they characterize the heartwood, not sapwood. So the only option that is correct is the last one, option number D. Question number 23 is from the chapter Biomolecules, and the question is, a non-proteinaceous enzyme is, options are deoxyribonuclease, lysozyme, ribozyme, and ligase. So what is an enzyme? Enzyme is a biological catalyst. Okay, biological catalysts. That means they catalyze the biological reaction, or they speed up the biological reactions. Okay. Now we know that mostly the enzymes are proteinaceous in nature. That means they are made up of protein. But there is an exception, and that is ribozyme, which is non-proteinaceous enzyme. Okay. And others are proteinaceous enzyme. And what is ribozyme? It is basically an RNA molecule, and more specifically 23R RNA. And it acts as a catalyst during protein synthesis. Okay, it acts as a catalyst during protein synthesis. Okay, therefore option C is the correct answer. Question number 24 is from the chapter Cell, and the question is, select the mismatch. Okay, that means which is not correctly matched. The first one is, methanogens are matched with prokaryotes, and that is true because methanogens are biogas producing bacteria, biogas or methane producing bacteria, because biogas contains methane. Okay, producing bacteria, and bacteria are prokaryotes, so this is a correct match. Okay, also these are type of archaebacteria. Now the second one is gas vacuoles, and it is matched with green bacteria. So this is also a correct match because in green bacteria gas vacuoles are found. Okay. Then the third one is large central vacuole, and it is matched with animal cells. This is not correct because large central vacuole is a feature of plant cell, not animal cell. Okay. In animals, the vacuoles are either absent, or if they are present, they are very small. Okay, so this is not a correct match. And the last one, protists are matched with eukaryotes. Yes, this is true. We know that protists are eukaryotic in nature. They have a well-defined nucleus along with nuclear membrane. Okay, so the only mismatch here is the option number C. So this is the correct answer. Question number 25 is from the chapter Cell, the Unit of Life, and the question is select the wrong statement. Okay, so we have to find the incorrect one. The first statement says mycoplasma is a wall-less microorganism. Yes, this is a correct statement. Okay, it does not have a cell wall. Then we come to the second statement. It says bacterial cell wall is made up of peptidoglycan. This is also true. Okay. Third statement says pili and fimbriae are mainly involved in motility of the bacterial cells. This is not true. Okay. See, bacterial cells have different types of appendages. For example, we have flagella, sometimes cilia also. Then we have pili and fimbriae. Okay. Now, what is the role of flagella? It helps in motility of the bacterial cell. In case of flagella, flagella also helps in motility. But pili and fimbriae don't play a role in motility. What is the role of pili? It helps in conjugation. Okay, it forms conjugation tube. Conjugation tube during the process of conjugation. Okay, whereas if we talk about the fimbriae, it provides attachment. It helps in attachment of the bacterial cell to the substratum or the host cell. Okay, so this statement is incorrect because pili and fimbriae don't play a role in motility of the bacterial cell. Instead, flagella play a role in motility. Okay, then the last statement says cyanobacteria lack flagellated cells. This is also correct. Cyanobacteria are non-motile, therefore they lack flagellated cells because flagella is not required there. Okay, so the only wrong statement is option C. So this is the correct answer. Question number 26 is again from the chapter Cell, and the question is a cell organelle containing hydrolytic enzymes is. Options are mesosome, lysosome, microsome and ribosome. So what are hydrolytic enzymes? These are the enzymes that help in the breakdown of the macromolecules or biomolecules. Okay, so this way they help in their digestion. So we can say that hydrolytic enzymes help in breakdown or digestion of biomolecules. Okay. Let's see the examples. There are some well-known hydrolytic enzymes such as proteases, and we know that they help in the digestion of proteins, right? Similarly, nucleases, they help in the digestion of nucleic acid. Now, these hydrolytic enzymes are found in which organelle of the cell? In none other than lysosome. Okay, this one. And therefore, sometimes lysosome also digests the complete cell. And this is the reason why they are known as the suicide bags of the cell. Okay, suicide bags of the cells. Okay, because they can digest the complete cell sometimes, but not always, right? So, therefore, the correct answer is option number B, lysosome. Question number 27 is from the chapter Cell Cycle and Cell Division, and the question is during cell growth DNA synthesis takes place in. Options are M phase, S phase, G1 phase and G2 phase. Okay, so let's first understand the events of a cell cycle. Cell cycle mainly consists of two phases. First one is the interphase, and the second one is M phase or the mitosis phase. Okay, so during the M phase, cell division takes place. Cell division or the mitosis of the cell takes place. Whereas interphase is the resting phase of the cell. Resting phase or the phase between two M phases. Okay, two cell divisions. Now, this interphase again consists of three phases. First one is G1 phase, second one is S phase, and third one is G2 phase. Okay, now G1 phase means growth one phase, G for growth and S for synthesis. So during the G1 phase, the cell is metabolically active. Cell is metabolically active and it grows continuously. And the most important thing is that during this phase only DNA polymerase enzyme is synthesized. Okay, DNA polymerase enzyme is synthesized. Now this enzyme is the one that is responsible for the duplication of DNA. But the thing is that this enzyme does not get activated during G1 phase. It is only synthesized during G1 phase, and it gets activated in S phase or the synthesis phase, and due to this reason DNA synthesis takes place during S phase, okay, with the help of DNA polymerase enzyme, right? Therefore, the correct answer will be option number B, S phase. What happens in G2 phase? Here the further growth of cell takes place, further growth. Then translation takes place, that means the formation of protein and other organelles also takes place, translation and formation of organelles. Then reorganization of the cell components take place, reorganization of cell components, so that the cell can be prepared for the next M phase. Okay, so according to this question, our answer will be option B. DNA synthesis takes place during S phase. Question number 28 is from the chapter Respiration and Plants, and the question is, which of the following biomolecules is common to respiration mediated breakdown of fats, carbohydrates and proteins? Options are acetyl coenzyme A, glucose 6-phosphate, fructose 1,6-bisphosphate and pyruvic acid. So let's see, if we talk about the breakdown of fats, we know that fats first break down into fatty acids and glycerol, right? Fatty acid and glycerol. Now through beta oxidation, these fatty acids and glycerols are converted into acetyl coenzyme A. Okay, acetyl coenzyme A. Now let's talk about the breakdown of carbohydrates, for example glucose. So we know that during glycolysis, glucose is converted into pyruvate, and then this pyruvate is converted into acetyl coenzyme A. Okay, then the next one is proteins, right? So we know that proteins will first break down into the amino acids, and then these amino acids get converted into pyruvate, and again pyruvate get converted into acetyl coenzyme A. So we can see that among these three biomolecules, fats, glucose and proteins, the common molecule is acetyl coenzyme A. Okay, therefore the correct answer will be option number A, acetyl coenzyme A. Question number 29 is from the chapter Transport in Plants, and the question is, a few drops of sap were collected by cutting across a plant stem by a suitable method. The sap was tested chemically. Which one of the following test results indicates that it is a phloem sap? Options are absence of sugar, acidic, alkaline and low refractive index. Okay, so we know that normally various types of pressures exist inside the plant. For example, root pressure, then we have phloem pressure, then there are some local pressures, okay, etc. And these pressures maintain the normal functioning of the plant, right? But when we cut the stem, okay, or cut any part, then these pressures are disturbed, okay. So on cutting the stem, above pressures are disturbed, right? And this will lead to the pumping nature. Okay, pumping will take place, and what will come out? Companion cells will come out. Okay, companion cells, and these companion cells will contain the proton. Okay, so along with the companion cells, protons will come out. So due to the presence of proton in the outside environment, the environment will become more alkaline. Okay, so this will lead to alkaline environment. So this will prove that it is a phloem sap. Therefore, the correct option will be option number C, alkaline. Okay. Question number 30 is from the chapter Plant Growth and Development, and the question is, you are given a tissue with its potential for differentiation in an artificial culture. Which of the following pairs of hormones would you add to the medium to secure shoots as well as Roots. Options are gibberellin and abscisic acid, indole 3-acetic acid and gibberellin, auxin and cytokinin, and auxin and abscisic acid. Okay, so if we talk about the hormone auxin, we know that it is responsible for the apical dominance, right? Apical dominance. That means it increases the length of roots and shoots. Okay, so it promotes lengthening, promotes lengthening of roots and shoots. Also, it coordinates with other growth hormones. Okay, now the second one, that is cytokinin. It promotes cell division. Okay, it is responsible for cell division, right? Now, the lower concentration of these two hormones, lower concentration of auxin and cytokinin, auxin and cytokinin helps in the shoot growth. Okay, growth of shoot. Whereas in higher concentration, they help in the growth of roots. Okay, higher concentration of these two hormones help in root growth. So we can say that these two hormones are required to secure shoots as well as roots. So option C will be the correct answer. Question number 31 is again from plant growth and development, and the question is phytochrome is a, options are chromoprotein, flavoprotein, glycoprotein and lipoprotein. So the correct answer is option A, that is chromoprotein. Okay, phytochrome is nothing but a chromoprotein, and we know that chromo means color, right? So this is a colored pigment, colored pigment in the plant, because this protein consists of a pigmented prosthetic group. Okay, and it helps in the perception of light, perception of light by the plant. Okay, and in turn it helps in various activities of the plant such as seed germination, seed germination, then flowering, etc. Okay, so the correct answer is option A, chromoprotein. Question number 32 is from the chapter Mineral Nutrition, and the question is which is essential for the growth of root tip? Options are manganese, zinc, iron and calcium, and the correct answer is option D, calcium. Okay, so we know that the tissues responsible for the division and differentiation of roots and shoots are meristematic tissues, right? Meristematic tissues. And the mineral responsible for their dividing activity is calcium. And this way the calcium helps in the growth of root tip. And where else is calcium used? It is used in the new cell wall synthesis. Okay, cell wall synthesis, and especially the middle lamellae, particularly the middle lamellae. We know that middle lamellae is made up of calcium pectate, right? Then it is also used in mitotic spindle during the cell division. Okay, so our correct answer is option number D. Question number 33 is from the chapter Photosynthesis in Higher Plants, and the question is the process which makes major difference between C3 and C4 plants is, options are respiration, glycolysis, Calvin cycle and photorespiration, and the correct answer is option D, that is photorespiration. So what is photorespiration? It is the inhibition of photosynthesis by high oxygen level. Okay, high oxygen level. See what happens during photosynthesis. During photosynthesis, we know that carbon dioxide is taken, okay, is taken in by the plants, and oxygen is given out. It is given out. But when there is high oxygen level in the environment, then plants start taking up oxygen. Okay, so oxygen is taken up by the plants and carbon dioxide is given out. So this is just the contrary of the photosynthesis, right? So it inhibits the photosynthesis. So inhibition of photosynthesis takes place due to increased level of oxygen, and this is known as photorespiration. Now it was observed that photorespiration mostly occurred in C3 plants. Okay, mostly in C3 plants as compared to the C4 plants. It was hardly observed in C4 plants because C4 plants were not affected by varying the oxygen level. Okay, so the major difference between C3 and C4 is this process, that is photorespiration. So option D will be the correct answer. Question number 34 is from the chapter Reproduction in Organisms, and the question is, which one of the following statements is not correct? So the first statement says water hyacinth growing in standing water drains oxygen from water that leads to death of fishes. So this is absolutely correct statement. Second statement says offspring produced by asexual reproduction are called clone. This is also correct. Why are they called clone? Because in case of asexual reproduction, no gamete fertilization takes place. Okay, no gamete fertilization, or simply no fertilization, because there is only one parent and therefore there is no variation in the offsprings. Okay, no variation in offsprings. So these are genetically similar to each other and therefore these are known as clone. Then the third statement says microscopic motile asexual reproductive structures are called zoospores. So yes, this is a correct definition of zoospores. Okay. Then the last statement says in potato, banana and ginger, the plantlets arise from the internodes present in the modified stem. No, this is not correct because plantlets always arise from nodes, not from internodes. What are the vegetative propagules in these plants? Vegetative propagules. So in case of potato, bud is the vegetative propagule. In case of banana, inflorescence is the vegetative propagule. Inflorescence. And in case of ginger, it is rhizome. Okay. So these are the parts through which the propagation takes place. And these represent the node of the plant, not internodes. So the last statement is incorrect. So this is the correct answer, option number D. Question number 35 is again from the chapter Reproduction in Organisms, and the question is, which one of the following generates new genetic combinations leading to variation? Options are nuclear polyembryony, vegetative reproduction, parthenogenesis and sexual reproduction. So it is quite obvious that the answer will be sexual reproduction because it involves the fusion of gametes from two different parents. Okay, so sexual reproduction involves two parents. One is male and the other is female. Okay, so male produces the male gamete and the female produces the female gamete. Okay, and then the fusion of these two gametes takes place. But before that, we know that these gametes are produced by the process known as meiosis, right? And this meiosis involves one of the steps that is known as recombination. Okay, and this recombination is majorly involved in the variation. Okay, and after that the fusion takes place, that is the fertilization. This is also responsible for the variation. So overall the progeny that is produced varies from both the parents. Okay, therefore the correct answer will be option number D, sexual reproduction. Question number 36 is taken from different chapters of biology, and here we have to match column 1 with column 2. So the first one in column 1 is pistils fused together. Okay, so what are pistils? These are nothing but the female reproductive part of the flower. Okay, female reproductive part of the flower or female reproductive organ of the flower. Okay, and they can contain one or more carpels. Now what happens when the carpels are fused together? Then this condition is known as syncarpous condition. Okay, because carpels are fused together. Whereas when carpels are not fused together, then that condition is known as apocarpus. So here it is asked about the pistils fused together. That means carpels fused together, and the correct match for this will be syncarpus. Okay, so the correct match for A will be number 3. Then the second one is formation of gametes. So we know that the process of formation of gametes is known as gametogenesis. That means formation of or genesis of gametes. Okay, so the correct match for B will be number 1. Then the third one is hyphae of higher ascomycetes. Ascomycetes are the fungi, right? And hyphae of higher ascomycetes are dikaryotic. So the correct match for C will be number 4. What is meant by dikaryotic? It means that one cell contains two genetically distinct nuclei. Okay, so two genetically distinct nuclei in one cell. This condition is known as dikaryotic. Okay. Now let's come to the fourth one, unisexual female flower. That means only one sex is there. Okay, and that is female. So it will be pistillate in nature because only pistil is present in unisexual female flowers and no stamen is present. Okay, so it will be matched with pistillate. So the correct match for D will be number 2. So according to this, the correct option will be option number A. Okay. Question number 37 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, in majority of angiosperms, options are a small central cell is present in the embryo sac. Now this is incorrect because the central cell present in the embryo sac is large, not small. Okay, so there is a large central cell in the embryo sac. Then the second statement says egg has a filiform apparatus. This is also incorrect because synergids have filiform apparatus. Okay, synergids which are present in the egg cell have filiform apparatus. Then the third one is there are numerous antipodal cells. This is also incorrect because the number of antipodal cells is only 3. Okay, there are only 3 antipodal cells, not numerous. Then the last statement says reduction division occurs in the megaspore mother cell. Yes, this is a correct statement. Okay, we know that megasporangium, this is a structure which has megaspores. Okay, so initially the megasporangium consists of megaspore mother cells, that is MMC in short, and these MMCs undergo reduction division, that is meiosis. Okay, so reduction division, that means meiotic division. And after meiotic division or the reduction division, MMC develop into megaspores. Okay, so this statement is correct, so option D will be the correct answer. Question number 38 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, pollination in water hyacinth and water lily is brought about by the agency of, options are bats, water, insects or wind, and birds. Okay, so as the name suggests, water hyacinth and water lily are aquatic plants, that means hydrophytes. Okay, hydrophytes. First one is water hyacinth and the second one is water lily. Now the interesting thing is that despite being hydrophytes, these are not pollinated by water. So we can say that not all hydrophytes are pollinated by water. Okay, so how are these pollinated? If we talk about the water hyacinth, we know that these contain very beautiful and conspicuous flowers. Okay, conspicuous flower, and they have violet or lavender colored flower. So these flowers are clearly visible to the insects. Okay, insects are attracted towards these flowers, and therefore insects act as the pollinating agent in case of water hyacinth. So what kind of pollination takes place? Entomophily. Okay, entomophily, that means pollination by insects. Okay, now if we talk about the second one, that is water lily, so it also has beautiful flowers, beautiful pinkish flowers. Okay, pinkish flower. So again, insects are attracted towards these flowers. So one type of pollination that is possible in water lily is entomophily, and at the same time they have large number of stamens. Okay, large number of stamens. That means large number of pollen grains will be released. Okay, large number of pollens will be released, and these pollens are pollinated by the wind. Okay, these are light, so they are pollinated by wind. So this is anemophily. Anemophily, that means pollination by wind. So we can say that in these two flowers, pollination can take place through insects as well as winds. Therefore, the correct answer will be option number C, insects or winds. Question number 39 is from the chapter Sexual Reproduction in Flowering Plants, and the question is, the ovule of an angiosperm is technically equivalent to, options are megaspore, megasporangium, megasporophyll and megaspore mother cell. So what is an ovule? Ovule is the structure that is found inside the ovary, right? So found inside the ovary, and after fertilization they develop into the seed. Okay, so they become seed after fertilization. Now inside the ovule, megaspore mother cells, that is MMC, are present, which undergo meiotic divisions, okay, or meiosis, to form megaspores. Okay, so here we have to find the structure that is equivalent to the ovule. That means inside that structure, megaspore mother cell should be present, and that should develop into the megaspores. Okay, so let's see. The first option is megaspore. So megaspore cannot be the correct answer because megaspore develops inside the ovule, right, after the meiotic division of MMC. So these are the structures which are found inside the structures that are known as megasporangium. Okay, so megasporangium is the structure inside which megaspores are found. Okay, then the third one is megasporophyll. Now phyll means leaves, so these are the leaves which bear megasporangium. Okay, which bear megasporangium, and then inside these megasporangium, megaspores are formed. Okay, and what is megaspore mother cell? These are the cells present inside the megasporangium which undergo meiotic division to form megaspores. Okay, so these are initially diploid cells, that is 2N cells, which undergo meiotic divisions to form megaspores. Okay, and these are found inside the megasporangium. So we have seen that megaspores are also found inside the megasporangium. So actually these megaspores were initially in the form of megaspore mother cell, but after meiotic division they become megaspores. Okay, and these are present inside the megasporangium, and here we saw that these are present inside the ovule. That means the ovule is technically equivalent to the megasporangium. Therefore option B will be the correct answer. Question number 40 is from the chapter Molecular Basis of Inheritance, and the question is, Taylor conducted the experiments to prove semi-conservative mode of chromosome replication on, options are E. coli, Vinca rosea, Vicia faba and Drosophila melanogaster. Okay, this is Vicia faba, not Vida faba. And the correct answer is option C, that is Vicia faba. Okay, so what is Vicia faba? This is commonly known as broad bean or faba bean, and it belongs to the family Fabaceae. Okay, it is a leguminous plant. Okay, and which part of this plant was taken for experiment? The root tip. So Taylor treated the root tip cells of the Vicia faba with radioactive thymidine. Radioactive thymidine to label the DNA, and then grew them in the normal medium to demonstrate semi-conservative mode of chromosome replication. Okay, and this was not the first time when this kind of experiment was conducted, because semi-conservative mode of chromosome was already proved by Messelson and Stahl first time on E. coli. Okay, so it was done by Messelson and Stahl in 1958, and then after that Taylor conducted this experiment. Okay, so since the question is asked about the Taylor, so the correct answer is option number C. Question number 41 is from the chapter Principles of Inheritance and Variation, and the question is the mechanism that causes a gene to move from one linkage group to another is called. Options are crossing over, inversion, duplication and translocation. So what is a linkage group? A linkage group represent all of the genes on a single chromosome. Okay, all of the gene on a single chromosome. And why they are known as linkage group? Because these are inherited as a group. Okay, these are inherited as a group and not individually. Therefore, these are known as linkage group. Now let's understand the meaning of the terms given here. First one is crossing over. So what is crossing over? It is the exchange of DNA between the homologous pair of chromosomes. Okay, so let's see here. Suppose this is one chromosome, this is another. So these are homologous pair of chromosomes. Okay, now how will crossing over take place? These two will attach to each other like this. Okay, so one part will attach and through this part the DNA will get exchanged. Okay, so exchange of DNA between homologous chromosomes is known as crossing over. Okay, now let's understand the second term, that is inversion. So inversion means the arrangement in the reverse order. Okay, so it is a kind of defect in which a segment of chromosome breaks off and reattaches itself in the reverse direction. Okay, for example, if this is a chromosome, here we have a segment AB. Okay, so in case of inversion, it will first break off from this position and then reattach itself at the same position but in the reverse order. That means A will come here and B will come here. Okay, so this is known as inversion. Inversion. Then the third one is duplication. Duplication. So as the name suggests, the DNA will get duplicated. That means multiple copies of DNA will be produced. Okay, multiple copies of DNA are produced. Okay, for example, if this is a chromosome, this is another, these are homologous pair of chromosomes. Okay, and each chromosome contain a segment AB. Okay, now what will happen? This segment from one chromosome will break off and it will attach on the second one. Okay, so the second one was already having an AB segment and it will get added. Okay, one more segment will get added here. So the segment will be duplicated. So this is known as duplication. Okay, and the last one is translocation. So what is translocation? Let's see. See, in the above cases, we saw that the DNA was transferred to the other chromosome, but within the same homologous pair. Okay, but in this case, translocation, the DNA is transferred to some another chromosome. Okay, that is not the part of this homologous pair. Okay, so these are the homologous pair of chromosomes. Here we have a DNA segment AB. Now this is not transferred to this one, but to some another one. Okay, some other chromosome, that is non-homologous chromosome. Okay, so we can see that this is known as translocation. So the DNA is translocated to a non-homologous chromosome. Okay, so here the question was asked that the mechanism that causes a gene to move from one linkage group to another, that means one chromosome to another. Okay, so here we are talking about the translocation, that is option number D is the correct answer. Okay, Question number 42 is from the chapter Molecular Basis of Inheritance, and the question is the equivalent of a structural gene is. Options are recon, muton, cistron and operon. So what is a gene? It is the basic unit of heredity. Okay, basic unit of heredity that is passed from parent to child. Okay, and it is made up of sequences of DNA. Made up of sequences of DNA, and it codes for a protein. Okay, codes for proteins. Now, the correct answer is option number C, that is cistron, because cistron is the coding part of the DNA. Okay, cistron is coding part of DNA. And the structural gene is also the part of DNA that codes for proteins. So it can be said that this structural gene is equivalent to cistron. So option C is the correct answer. Question number 43 is from the chapter Strategies for Enhancement in Food Production. And the question is, a true breeding plant is option A, always homozygous recessive in its genetic constitution. Option B, one that is able to breed on its own. Option C, produced due to cross pollination among unrelated plants. And the last one is near homozygous and produces offspring of its kind. So what is a true breeding plant? See, true breeding plant is developed after continuous self-pollination. Okay, continuous self-pollination. That means not any other type of plant is involved. Okay, and due to the continuous self-pollination, homozygosity is there. Okay, so the development of homozygous plant take place. Homozygous plant. So in homozygous, what will happen? The offspring will be of the same kind as the parent plant. Okay, so the correct option will be option number D, near homozygous and produces offspring of its kind. So option D is the correct answer. Question number 44 is from the chapter Molecular Basis of Inheritance, and the question is, which of the following rRNA acts as a structural RNA as well as ribozyme in bacteria? Options are 5.8S rRNA, 5S rRNA, 18S rRNA and 23S rRNA. And the correct answer is option number D, that is 23S rRNA. Okay, this is a direct question. It acts as structural RNA as well as ribozyme in bacteria, that is prokaryotes. And what is ribozyme? It is the RNA that acts as an enzyme. Okay, that acts as an enzyme. So this 23S rRNA also acts as enzyme during the process of translation in bacteria. Okay. Question number 45 is from the chapter Microbes in Human Welfare, and the question is, stirred tank bioreactors have been designed for, options are ensuring anaerobic conditions in the culture vessel, then purification of product, addition of preservatives to the product and availability of oxygen throughout the process. Okay, availability. Now, what are bioreactors? Stirred tank bioreactors. These are large vessels which are designed or which are developed in order to produce large number of microorganisms. Okay, to produce large number of microorganisms. And these stirred tank bioreactors have certain advantages. These have oxygen delivery system, so that oxygen is delivered continuously into the bioreactors. Okay, then it has temperature control system, and also pH control system, so that the condition is suitable for the growth of microorganisms. Okay, so the correct answer will be option number D, that is availability of oxygen throughout the process, because it has oxygen delivery system and it ensures that oxygen is continuously delivered into it. Okay, so option D will be the correct answer. Question number 46 is from the chapter Molecular Basis of Inheritance, and the question is, a molecule that can act as a genetic material must fulfill the traits given except. First option is, it should provide the scope for slow changes that are required for evolution. That is absolutely correct. The genetic material should have scope for evolution, right? Second statement says it should be able to express itself in the form of Mendelian characters. This is also a correct statement. Third one says it should be able to generate its replica. This is also correct. The genetic material should be self-replicating. Okay, self-replicating. And the last one says it should be unstable structurally and chemically. Now this is incorrect statement because it should be stable. Okay, if the genetic material is unstable, then what will happen? The expression will change, leading to loss in several metabolic functions, or we can say there will be inconsistency in the expression. Okay, inconsistency in expression, and that is not required here. So the molecule should be stable, structurally as well as chemically. So option D is the correct answer here. Question number 47 is again from the chapter Molecular Basis of Inheritance, and the question is, DNA dependent RNA polymerase catalyzes transcription on the strand of DNA which is called the. Options are anti-strand, template strand, coding strand and alpha strand. So what is transcription? It is the formation of RNA from double-stranded DNA. Okay, so DNA to RNA formation is known as transcription. And transcription always takes place in 5' to 3' direction, and the enzyme responsible for it is DNA dependent RNA polymerase. Okay, now suppose these are two strands of DNA. The above is 3' to 5' and the lower one is 5' to 3'. Okay, now we know that DNA dependent RNA polymerase enzyme will catalyze this transcription in the direction 5' to 3'. That means the above strand will be used. Okay, 5' to 3' like this, and this above strand is known as template strand, and the other one is known as coding strand. Okay, lower one will be known as coding strand, and the newly synthesized RNA will be similar to this coding strand. Okay, so here the strand that is used for RNA formation is the above one, that is template strand. So the correct answer will be option number B. Question number 48 is from the chapter Strategies for Enhancement in Food Production, and the question is, interspecific hybridization is the mating of. Option A, more closely related individuals within same breed for 4 to 6 generation, animals within same breed without having common ancestor, then two different related species, and the last one is superior males and females of different breeds. And the correct answer is option number C, that two different related breeds. So as the name suggests, interspecific, that means between two different species. Okay, two different species, but these two species should be related. They should not be totally unrelated. For example, we can take donkey and horse. So we know that these two belong to different species. Okay, different species, but they are related. So it is an example of interspecific hybridization. This is possible here. But if we take the example of donkey and lion, then this is not possible because they are totally unrelated. Okay. So the correct answer will be option number C, two different related species. Okay. Question number 49 is from the chapter Human Health and Disease, and the question is, which of the following is correct regarding AIDS causative HIV? So AIDS means acquired immunodeficiency syndrome, and it is caused by a virus known as human immunodeficiency virus, or HIV in short. Okay. Now, since it is a virus, we know that viruses have RNA as the genetic material. So HIV also has RNA as the genetic material. Okay. Now, this HIV will bring two identical molecules of RNA. Okay. Two identical single-stranded RNA molecules. Okay. Now, what will happen? This virus, that is HIV, will attack the macrophage cell of human body. Which cell? Macrophage. Now we know that HIV will bring RNA with it. So first these RNAs will get converted into DNA through a process known as reverse transcription. Reverse transcription. Because it is just the opposite of transcription. Okay. And the enzyme responsible for this conversion is reverse transcriptase enzyme. Now what will happen, as this macrophage cell will divide inside the human body, along with this cell, the DNA of HIV will also multiply, and in this way it will escape the human immune system. Okay, and this is the reason macrophage cell is also known as factory, HIV factory. Okay, now let's see the statements. The first statement says HIV does not escape but attacks the acquired immune response. No, this is incorrect, because we have seen that it escapes the immune response, because along with macrophage cell it keeps on dividing. Okay, so immune system is unable to prevent it from dividing. Now the second statement says HIV is enveloped virus containing one molecule of single stranded RNA and one molecule of reverse transcriptase. Now this is incorrect, because we have seen that it contains two identical single stranded RNA molecules. Okay, so two molecules of single stranded RNA, not one. Okay, also it will contain two molecules of reverse transcriptase enzyme. Okay, then the third statement says HIV is enveloped virus that contains two identical molecules of single stranded RNA and two molecules of reverse transcriptase. Yes, this is true. First of all, the HIV is enveloped, that you have to remember. Then it contains two identical single stranded RNA molecule and two molecules of reverse transcriptase enzyme. Okay, so this is a correct statement. Last one says HIV is unenveloped retrovirus. This is not true, because we have seen this is enveloped virus. Okay, so the only correct statement is statement C. So option C is the correct answer. Question number 50 is related to animal kingdom, and the question is, among the following edible fishes, which one is a marine fish having rich source of omega-3 fatty acids? Options are mackerel, mistus, mangoor and mrigala. So the correct answer is option number A, that is mackerel. This is the only marine fish given in the options. Others are freshwater fishes. Okay, freshwater fishes. And omega-3 fatty acid is found in marine fishes. And what is omega-3 fatty acids? Which kind of fatty acid is this? This is an example of polyunsaturated fatty acids. Okay, polyunsaturated fatty acid, and it is characterized by a double bond. Double bond. Now it is rich in vitamin B, and more particularly vitamin B12. Okay, this is found in abundant quantity. Other than that, proteins are also found in this. Okay, so the correct answer according to this question is option number A, mackerel. It contains omega-3 fatty acid. Question number 51 is from the chapter Microbes in Human Welfare, and here we have to match column 1 with column 2. Okay, so the first one in column 1 is citric acid, and we know that citric acid is produced by a fungus named Aspergillus niger. Okay, Aspergillus niger, and it is a fungus. So the correct match for A will be number 3. So option D is eliminated here, because it is not matched with number 3 here. Then the second one is cyclosporin A. So what is cyclosporin A? It is an immunosuppressive agent, and it is produced by a fungus named Trichoderma polysporum. Okay, Trichoderma polysporum. It is again a fungus. So the correct match for B will be number 1. So here option A is eliminated, because here B is matched with number 4. Okay. Now the third one is statins. What is statins? It is blood cholesterol lowering agent. Okay, and it is produced by an yeast, Monascus purpureus. This is an yeast. So yeast is also example of fungus only. This is another type of fungus. So the correct match for C will be number 4. That means option C will be the correct answer. Okay. Last one is butyric. It is produced by the bacteria named Clostridium butylicum. Okay. Clostridium butylicum, and it is a bacteria. So according to this, the correct option should be option number C. Question number 52 is from the chapter Environmental Issues, and the question is biochemical oxygen demand may not be good index for pollution for water bodies receiving effluents from. Options are sugar industry, domestic sewage, dairy industry and petroleum industry. So what is biochemical oxygen demand? It is the measurement of dissolved oxygen that is consumed by consumed by aerobic bacteria living on the organic material or growing on organic material in a water sample at a specific temperature over a specific time period. Okay, so simply we can say that it is the amount of dissolved oxygen that is required by aerobic microbes living in water. Okay, so higher biochemical oxygen demand, higher BOD represents highly polluted water. Okay. Now for industries, we know that the industries must release organic material as effluents, and for them it will be a good index for pollution. Okay, so let's see, in the first one we have sugar industry. So definitely sugar industry is going to release the organic material or the biodegradable material as the effluents. Okay, and same will be the case with domestic sewage and dairy industry. Okay, so these are going to release the biodegradable or organic materials as effluents, but the case is not true with petroleum industry. Okay, what is the effluent in petroleum industry? It will be inorganic substances. Okay, inorganic substances will be released from the petroleum industry, and especially it is rich in radium based toxic substances. Okay, so this is not going to favor the algal or plankton growth, and therefore it will not have any effect on the biochemical oxygen demand. So no effect on BOD. Therefore, for petroleum industry, BOD is not a good index for pollution. So option D is the correct answer. Question number 53 is from the chapter Organisms and Population, and the question is the principle of competitive exclusion was stated by. Options are Verhulst and Pearl, C. Darwin, G.F. Gauss and MacArthur. So the correct answer is option C, that is G.F. Gauss. Okay, so it was Gauss who gave the principle of competitive exclusion, and in that he stated that no two species which compete for the same limited resource can coexist at constant population values. Okay, constant population values. That means if there are limited resource in a region and two species are competing for the same resource, then they cannot coexist. Okay, and eventually what will happen? The competitively superior one will eliminate the inferior one. Okay, so competitively superior species will eliminate the competitively inferior one. Okay, and this is known as principle of competitive exclusion. So it will exclude the inferior one, okay, or eliminate the inferior one. So option C is the correct answer. Question number 54 is from the chapter Biodiversity and Conservation, and the question is, which of the following national parks is home to famous musk deer or hangul? Options are Dachigam National Park, Jammu and Kashmir; Kibul Lamjao National Park, Manipur; Bandhavgarh National Park, Madhya Pradesh; and Eagle Nest Wildlife Sanctuary, Arunachal Pradesh. Okay, so national park, we know that national parks are in situ methods of conservation. Okay, so here it is asked about the national park which is home to musk deer or hangul. So the correct answer will be option number A, Dachigam National Park, Jammu and Kashmir. Okay. Question number 55 is from the chapter Environmental Issues, and the question is, a lake which is rich in organic waste may result in option A, mortality of fish due to lack of oxygen; option B, increased population of aquatic organisms due to minerals; option C, drying of the lake due to algal bloom; and option D, increased population of fish due to lots of nutrients. So let's see, the lake is rich in organic waste. Okay, that means a large amount of organic waste is present in the lake, and that is going to increase the biochemical oxygen demand. Okay, biochemical oxygen demand will increase, and due to that, oxygen will be consumed in the lake faster, and it will result in lack of oxygen. Okay, lack of oxygen, that means lower amount of oxygen will be available to the fishes. Okay, and this will result in the mortality of fishes. Therefore option A will be the correct answer. Okay. Question number 56 is again from the chapter Environmental Issues, and the question is, the highest DDT concentration in aquatic food chain shall occur in. Options are eel, phytoplankton, seagull, and crab. Okay, so if we talk about DDT, we know that it is a harmful chemical, and the reason is that it is non-biodegradable. Okay, DDT is non-biodegradable substance. So it neither metabolizes nor it is excreted out. Okay, neither metabolizes nor excreted, and this makes DDT a harmful or toxic substance. Okay, so what happens? It gets accumulated. It gets accumulated, and the amount of DDT or any toxic substance in general goes on increasing from one trophic level to another, and this process is known as biomagnification. Biomagnification. Okay, so DDT will also undergo biomagnification. And here, if we talk about the aquatic food chain, we know that the transfer takes place in this manner. First, phytoplankton, which is a producer in the aquatic environment. Then phytoplanktons are eaten by zooplanktons. Zooplankton. These are primary consumers. Then small fishes, and then large fishes in general. Okay. Now if we arrange the given options in this manner, then the first one will be phytoplankton again. So phytoplankton cannot be the correct answer because this will have the minimum amount of DDT. Okay, it will go on increasing from the one trophic level to next. So first one is the phytoplankton. So definitely it's not going to be highest in phytoplankton. The next will be eel, because eel is a small fish. Okay, so eel will consume the phytoplankton. So the amount of DDT will increase from phytoplankton to eel, and then eel will be consumed by crab. So again the amount will increase, and finally crab will be consumed by seagull. Seagull, that means seagull will have highest amount of DDT. Highest amount of DDT. So the correct answer will be option number C, seagull. Question number 57 is from the chapter Human Health and Disease, and the question is, which of the following sets of diseases is caused by bacteria? Options are herpes and influenza, cholera and tetanus, typhoid and smallpox, and tetanus and mumps. Okay, so in the first one we have herpes and influenza, and both of them are caused by viruses. Okay, so both are viral diseases, so this is not the correct option. Then in the second one we have cholera and tetanus. So cholera is caused by Vibrio cholerae. Okay, Vibrio cholerae, and that is a bacteria. Okay, and the second one is tetanus. So tetanus is caused by Clostridium tetani. Clostridium tetani, and that is also a bacteria. So yes, both of them, that means this set of diseases is caused by bacteria. Okay, let's see other options as well. In the third one we have typhoid and smallpox. So typhoid is caused by Salmonella typhi. Salmonella typhi, and that is a bacteria. So first one is caused by bacteria, but the second one, that is smallpox, is caused by virus. Okay, so smallpox is a viral disease, therefore this is not the correct option again. And the last one we have tetanus and mumps. So we have seen that tetanus is caused by Clostridium tetani, that is a bacteria. Okay, but the second one, that is mumps, is a viral disease. Okay, and it is caused by variola virus. So the only set in which both the diseases are caused by bacteria is the second one. So option B will be the correct answer. Question number 58 is from the chapter Animal Kingdom. And the question is, match column 1 with column 2 for housefly classification and select the correct option using the codes given below. Okay, so housefly. So what is the scientific name of housefly? It is Musca domestica, right? Musca domestica. So here Musca is the name of the genus and domestica is the species, right? Now family of the housefly is Muscidae. Muscidae. Order is Diptera. Order is Diptera. Class is Insecta because it belongs to Insecta. It is an insect, right? And the phylum, we know that insects belong to Arthropoda phylum, so this is Arthropoda. Okay. So here first one is family. We know that family is Muscidae. So the match for A is number 3. Okay. So first option and last options are eliminated. Either B or C can be the correct answer. Then order is Diptera. That means the match for B will be number 1. So option number B. Okay. And the class is Insecta. So C4, and phylum Arthropoda. So the match for D will be 2. So yes, option B is the correct answer. Question number 59 is again from the chapter Animal Kingdom, and here we have to choose the correct statement. Okay, so the first statement says all Pisces have gills covered by an operculum. So Pisces means fishes, and we know that all the fishes respire through gills, and in some fishes the gills are covered by an operculum, that is also known as gill cover. Okay, but it is present only in some fishes, not in all fishes, so this is not correct. For example, in cartilaginous fishes, the operculum is absent. Then we come to the second statement. It says all mammals are viviparous. Viviparous means they give birth to young ones. They do not lay eggs, right? So in general, mammals are viviparous. But this is not always true because there are some exceptions. So this is also incorrect statement. For example, if we talk about ornithorhynchus, that means platypus, and echidna, these are exceptions because these are not viviparous, these are oviparous. Okay, therefore this statement is incorrect. Then we come to the third one. Third statement says all the cyclostomes do not possess jaws and paired fins. Yes, this is a correct statement because cyclostomes are jawless fishes. Okay, and these are also known as agnatha. Okay, so this is the correct one. The last one says all reptiles have a three-chambered heart. So in general, we know that reptiles have a three-chambered heart, but again there are exceptions. There is an exception actually, that is crocodile. Okay, crocodile. We know that crocodiles have a four-chambered heart. Four-chambered heart. So again, this statement is not always true. So the only correct statement is option number C. Question number 60 is from the chapter The Living World, and the question is, study the four statements A to D given below and select the two correct ones out of them. Okay, so four statements are given here and we have to find out the two correct statements. So the first statement says definition of biological species was given by Ernst Mayr. This is the correct statement. So according to Ernst Mayr, a species is a group of actually or potentially interbreeding natural population. Okay, interbreeding natural populations, and these are reproductively isolated from other such groups. That means all the organisms belonging to one species are able to interbreed. Okay, these are able to breed among themselves, but any organism of this species will not be able to breed with other organism that belongs to any other species. Okay, so these are reproductively isolated groups, right? Now we come to the second statement. It says photoperiod does not affect reproduction in plants. This is not true, because photoperiod is able to affect the reproduction in plants by regulating the flowering timing of the plants. Okay, so photoperiod, let's write it down here. Photoperiod can regulate the flowering timing of the plants, and this way they are able to affect the reproduction in plants. Okay, now we move to the third statement. It says binomial nomenclature system was given by R.H. Whittaker. Again, this is incorrect, because binomial nomenclature was given by Carolus Linnaeus. Carolus Linnaeus, and not R.H. Whittaker. R.H. Whittaker gave the five kingdom classification of the living organisms. Okay. Then the last statement says in unicellular organisms, reproduction is synonymous with growth. Okay, so yes, this is a correct statement, because if we take the example of amoeba, then we know that in case of reproduction, it will undergo binary fission and it will produce two amoeba, okay, from itself. So we can say that this amoeba has reproduced, or we can say that this amoeba has grown in number. Okay, so this way reproduction and growth is synonymous, right? So here the parent amoeba has changed into the daughter amoeba. Okay, but in case of sexual reproduction, what happens? The parent remains same. There is no effect on the parent, but here the parent itself has divided. Okay, so here the reproduction is synonymous to the growth. So which statements are correct? The first one and the last one. So A and D are correct. That means option D will be the correct answer. Question number 61 is from the chapter Structural Organization in Animals, and the question is, in male cockroaches, sperms are stored in which part of the reproductive system? Okay, options are vas deferens, seminal vesicles, mushroom glands and testis. And the correct answer is option B, that is seminal vesicles. Okay, what happens? The formation of sperms or spermatogenesis, that is the formation of sperms, takes place in the primary sex organs in cockroaches, and that is none other than testis. Okay, so it looks something like this. This is one testis, this is another. So the spermatogenesis takes place in these. That means sperms are formed in these testes. Okay, and then the sperms move through a duct known as vas deferens. So these are vas deferens, through which the sperms move out from the testis and reach the seminal vesicles. Okay, so here we have seminal vesicles. Now when the sperms reach in seminal vesicles, these are stored here till they are ejaculated outside the seminal vesicles. Okay, so storage takes place in the seminal vesicles. Therefore option B will be the correct answer. Question number 62 is again from the chapter Structural Organization in Animals, and the question is smooth muscles are. So what are smooth muscles? Let's understand. We know that muscles are of two types. First one is voluntary muscles and the second type is involuntary muscles. Okay. So as the name suggests, voluntary means these can be used according to our own will, or we can say these are under our control, whereas involuntary are not under our control. Okay. The example of voluntary muscles are skeletal muscle. That means the muscles attached to our skeletal system. So we can walk, we can stand up, we can lie down whenever we want. So these are voluntary. Whereas involuntary, there are two types of involuntary muscles. First one is cardiac muscle, that is found in our heart. Okay, heart. And the second one is smooth muscles, which are found in our internal organs. Internal organs, for example, urinary tract, reproductive tract, etc. Okay, well, so let's see how does skeletal muscle look like this. One second. It looks like this. It has striations like this. Okay, it has a striped appearance and these are known as striations, and these are multinucleate, so many nuclei are there. Now if I show you the cardiac muscles, these are branched like this. These are uninucleate, one nucleus is there, and it also has striped appearance. Okay, so there are striations in the cardiac muscles as well. You understand what is striations? Now smooth muscles, so it looks like this. Both the ends are tapering. Okay, tapering ends. It is also uninucleate, one nucleus is there, but there are no striations here. Okay, no striations as you can see, whereas skeletal muscle and cardiac muscles have striations. Now let's see. In the first option, it is given that smooth muscles are voluntary. So it is incorrect because we know that smooth muscles are example of involuntary muscles, right? So here only it is proved that this is incorrect. So let's cancel this option. Then we come to the second option. So these are involuntary, yes. Fusiform, yes. Fusiform means they have tapering ends like this. So we have seen that they have such structure. So this is also correct. And non-striated, that means they do not have striation. This is also correct. We have seen that striations are present in skeletal and cardiac, but not in smooth. So yes, this is correct. Okay. Third option says voluntary, again incorrect because smooth muscles are involuntary. Then the last option says involuntary, yes. Cylindrical, no, because we have seen skeletal as cylindrical, not smooth. Okay, and striated is also incorrect because these are non-striated. So the only correct option is option number B. Question number 63 is from the chapter Respiration in Plants, and the question is oxidative phosphorylation is option A, formation of ATP by energy released from electrons removed during substrate oxidation. Option B, formation of ATP by transfer of phosphate group from a substrate to ADP. Option C, oxidation of phosphate group in ATP, and option D, addition of phosphate group to ATP. Okay, so let's see. We know that phosphorylation means synthesis of ATP by the addition of phosphate group. Okay, so inorganic phosphate gets added to ADP in order to produce ATP. Okay, so this is simply phosphorylation. Now this process is driven by the energy, and that energy is released by oxidation of substrates. Okay, oxidation of substrates. And what are the substrates here? NADH plus H plus and FADH2. So these two are oxidized, and when these are oxidized, electrons are removed and these electrons are transferred to the electron transport chain that is located in the inner mitochondrial membrane. Okay, inner mitochondrial membrane. Now we know that electron transport chain consists of enzyme complexes. So total 5 enzyme complexes are there, and the electron has to move from one complex to another, and finally it is received by oxygen molecule. Okay, so oxygen is the final receiver. Now during the movement of electron from one complex to another, there is development of a gradient known as proton gradient, and this is responsible for the release of energy. Okay, so let's see which option should be the correct one. The first option says formation of ATP by energy released from electrons removed during substrate oxidation. So yes, we have seen that these substrates, that is NADH and NADH plus H plus and FADH2, are oxidized, and from that electrons are removed, and that is responsible for the release of energy. Okay, so this option will be the correct answer. Question number 64 is from the chapter Biomolecules, and the question is, which of the following is least likely to be involved in stabilizing the three-dimensional folding of most proteins? Options are ester bonds, hydrogen bonds, electrostatic interaction, and hydrophobic interaction. So the correct answer will be option A, that is ester bonds, because we know that hydrogen bonds, electrostatic interaction, and hydrophobic interaction will be present in proteins along with the peptide bonds. Okay, whereas ester bond is not present in proteins. Okay, not present in proteins, or not formed in proteins, because ester bonds are formed in nucleic acids and lipids. Okay, formed in nucleic acids and lipids, but not in proteins. Okay, therefore option A is the correct answer. Question number 65 is again from the chapter Biomolecules, and the question is, which of the following describes the given graph correctly? Okay, so in the graph we can see that there is potential energy of a reaction that is represented here. Okay, so we can see that there are two graphs of the same reaction, that means different conditions are considered, and basically it is related to the enzyme. Okay, the function of an enzyme, and what does an enzyme do? It reduces the activation energy. Okay, it reduces the activation energy of a reaction. So here A represents the activation energy of the lower graph and B represents the activation energy of the upper graph. Okay, now we know that an enzyme reduces the activation energy. That means A must be the activation energy in the presence of an enzyme, right? So A represents the activation energy in the presence of enzyme, in the presence of enzyme, because the energy has been reduced in this case, whereas B represents the activation energy in the absence of enzyme, because here the energy is higher. Okay, absence of enzyme, right? Now we can see that substrate has a lower potential energy, the higher potential energy. Okay, because on y-axis potential energy is shown, and product has a very low potential energy, right? So the potential energy of substrate is higher than the potential energy of product. That means there is a loss of energy. Okay, or we can say energy has been released. So energy is released, and energy is released, that means the process is exothermic. Exothermic. Okay, now let's see the options. In the first option it is said that there is exothermic reaction. Yes, there is exothermic reaction with energy A in absence of enzyme and B in the presence of enzyme. This is not correct, because we have seen that A is lower. Okay, that means A is the energy in the presence of enzyme, right? Not in absence of enzyme. Let's move to the second statement. It says endothermic. Now this is incorrect, because we have seen that the process is exothermic. Then we move to the third statement. It says exothermic reaction, yes, with energy A in presence of enzyme and B in the absence of enzyme. Yes, this is a correct statement. Okay, the last statement says endothermic. Again, this is incorrect, because the process is exothermic. So the only correct option is option C. So this is the correct answer. Question number 66 is from the chapter Cell Cycle and Cell Division, and the question is, when cell has stalled DNA replication fork, which checkpoint should be predominantly activated? Option A, both G2M and M. Option B, G1S. Option C, G2M. And option D, M. Okay. So let's see. We know that cell cycle mainly consists of an interphase and an M phase, that is mitosis phase. Okay. This is M phase. Now during mitosis, the actual cell division takes place, whereas interphase is also known as the resting phase of the cell, and during this phase cell prepares itself for the cell division or the mitosis. Okay, now this interphase further consists of G1 phase, then S phase, and then G2 phase. Okay, G1 means growth 1 and G2 means growth 2. So the growth of cells take place during G1 and G2, whereas during S phase DNA replication takes place. DNA replication. Now the question says that the cell has stalled DNA replication fork. Stalled means stopped working. Okay, stopped. So this means that the process of cell division has stopped during this S phase. Okay, so the process has not passed S phase, so the checkpoint also must be present prior to this S phase, not after S phase. Okay, so wherever G2 or M is present, this is not the correct answer. So option C and D is eliminated because the process has not crossed S phase. So how can it reach G2 or M phase? Okay, so the correct answer will be, okay, option A is also eliminated. So only option left is B. So the checkpoint must be G1S. That means between G1 and S. Okay. So option B is the correct answer. Question number 67 is again from the chapter Cell Cycle and Cell Division. And the question is, match the stages of meiosis in column 1 to their characteristic features in column 2 and select the correct option using the codes given below. So the first stage that is mentioned in column 1 is packetine. So what happens during packetine? During packetine, crossing over takes place. Okay. So packetine, crossing over of the chromosomes take place. So the correct match for A will be number 3. So here the only option in which A is matched with 3 is option number B. Okay, so this will be the correct answer. Still we'll see other options as well, other matches as well. Second one is metaphase 1. So what happens during metaphase? During metaphase, the chromosomes align themselves at the equatorial plate. Okay, like this. So chromosomes align at the equatorial plate during the metaphase. Metaphase. Alignment of chromosomes at the equatorial plate. So the correct match for B will be number 4. Okay, yes, this is also matched. Then diakinesis. So what happens during diakinesis? Terminalization of the chiasmata. So option C will be matched with this third one will be matched with the second in column 2. Okay. So B will be matched with number 4 and C will be matched with number 2. And the last one is zygotine. Okay, it is zygotine. So it will be matched with pairing of homologous chromosomes. Okay, during zygotine stage, pairing of homologous chromosomes takes place. So it is confirmed that option B is the correct answer. Question number 68 is from the chapter Digestion and Absorption. And the question is, which hormones do stimulate the production of pancreatic juice and bicarbonate? Options are insulin and glucagon, angiotensin and epinephrine, gastrin and insulin, and cholecystokinin and secretin. So the correct answer will be option number D, cholecystokinin and secretin. So what is the function of cholecystokinin? So basically this is a hormone which is produced in our small intestine. Okay. And it has a fundamental role in the digestive process. When fats and proteins enter our small intestine, this hormone triggers the gallbladder and pancreas to contract. And this way it helps in the release of bile from gallbladder and pancreatic juice from the pancreas. Okay. Pancreatic juice from pancreas. So first one is done. The question says which hormones stimulate the production of pancreatic juice. So yes, this cholecystokinin helps in the production of pancreatic juice. Now the second one, that is secretin. So what are the functions of secretin? It has mainly three functions. Okay. First is the regulation of gastric acid. Regulation of gastric acid. Then regulation of pancreatic bicarbonate. Pancreatic bicarbonate, and then osmoregulation. Okay, osmoregulation. So these are the functions of secretin. So we can see that this helps in the production of bicarbonate. So option D will be the correct answer. Question number 69 is from the chapter Breathing and Exchange of Gases, and the question is the partial pressure of oxygen in the alveoli of the lungs is option A less than that of carbon dioxide, option B equal to that in the blood, option C more than that in the blood, and option D less than that in the blood. Okay, so let's talk about the alveoli first. If we talk about alveoli, then here the partial pressure of oxygen is 104 and that of carbon dioxide is 40. Okay, and if we talk about the blood, deoxygenated blood, okay, deoxygenated blood, so here the partial pressure of oxygen is 40 and the unit is millimeter of mercury. Okay, and that of carbon dioxide is 45. Now in oxygenated blood, if we write the partial pressure of oxygen, it is 95. Okay, now let's see the options. In the first option, it is said that it is less than that of carbon dioxide. So we can see that partial pressure of oxygen in the alveoli is not less than that in carbon dioxide. It is more than that of carbon dioxide. Okay, so this is incorrect statement. Then the second statement says equal to that in the blood. So in the alveoli, it is 104 partial pressure of oxygen. In the blood, it is 40 and 95. Okay, for deoxygenated it is 40, for oxygenated it is 95. So it in both the cases it is more than that in the blood. Okay, so not equal to and not less than that in the blood, it is more than that in the blood, whether oxygenated or deoxygenated. Okay, so option C will be the correct answer. Question number 70 is from the chapter Neural Control and Coordination, and here we have to choose the correct statement. Okay, so the first statement says receptors do not produce graded potentials. Now this is not correct because receptors produce graded potentials. Now what are graded potentials? These are short distance potentials. Okay, short distance potentials are graded potentials, unlike the action potential. Okay, so remember that action potential are long distance potentials. So here the receptors produce the short distance potential that are graded potential. So this is incorrect statement. Now second statement says nociceptors respond to changes in pressure. This is also incorrect statement because nociceptors respond to changes in pain. Okay, these are sensitive to pain, not pressure. Okay, then the third statement says Meissner's corpuscles are thermoreceptors. This is also incorrect. So these are not thermoreceptors, these are mechanoreceptors. Okay, mechanoreceptors, and these are sensitive to fine touch. Okay, sensitive to touch. Then the last statement says photoreceptors in the human eye are depolarized during darkness and become hyperpolarized in response to light stimulus. Yes, this is the correct statement. Okay, so option D will be the correct answer. Question number 71 is from the chapter Chemical Coordination and Integration, and the question is Graves' disease is caused due to, options are hypersecretion of adrenal gland, hyposecretion of thyroid gland, hypersecretion of thyroid gland, and hyposecretion of adrenal gland. Okay, so what is Graves' disease? It is an autoimmune disease, autoimmune disease or autoimmune disorder. Okay, That means the body is unable to differentiate between the self and non-self cells, or the immune system of our body is unable to do so, and due to that it attacks its own cells. Okay, so in Graves' disease the immune system attacks the thyroid gland of the body. Okay, in Graves' disease immune system attacks the thyroid gland of the body, causing it to make more thyroid hormones than the body needs. Okay, so it leads to overproduction of the thyroid hormones, overproduction of thyroid hormones. So due to this, many of the body functions speed up. Okay, many of the body functions such as metabolism speeds up. Okay, so it is caused due to overproduction of thyroid hormones. That means hyper secretion of the thyroid glands. Okay, so option C will be the correct answer. Now if we talk about the symptoms of Graves' disease, so it has many symptoms such as weight loss, then diarrhea, then tremors, and the most important one is protruding eyes. Okay, protruding eyes. And this disease is characterized by exophthalmic goiter. Okay, so option C is the correct answer here. Question number 72 is from the chapter Locomotion and Movement, and the question is, name the ion responsible for unmasking of active sites for myosin for cross bridge activity during muscle contraction. Options are potassium, calcium, magnesium and sodium. So we know that the actin filament has myosin binding sites on it. Okay, myosin binding sites or the active sites. Okay, now these sites are covered or masked by unit of troponin. Okay, masked by troponin unit. Okay, now when myosin has to bind there, then this troponin has to be removed from there, and that is done with the help of calcium ion. Therefore the correct answer will be option number B, calcium. Okay, question number 73 is from the chapter Body Fluids and Circulation, and the question is, name the blood cells whose reduction in number can cause clotting disorder leading to excessive loss of blood from the body. Options are thrombocytes, erythrocytes, leukocytes and neutrophils, and the correct answer is option A, that is thrombocytes. Okay, so we know that platelets are responsible for the clotting of blood in our body. Another name of platelets is thrombocyte. Okay, so option A is the correct answer. Erythrocytes are the red blood cells. Okay, RBCs or red blood cells which contain pigments known as hemoglobin, and this hemoglobin is responsible for transporting oxygen to various parts of our body. Okay, then the third one is leukocytes. So this is a type of WBC, that is white blood cell. And the last one is neutrophil. It is also a type of white blood cell that is responsible for the immunity in our body. Okay, so according to this question, the correct answer is the first one, option number A. Question number 74 is from the chapter Chemical Coordination and Integration, and the question is, name a peptide hormone which acts mainly on hepatocytes, adipocytes and enhances the cellular glucose uptake and utilization. Options are gastrin, insulin, glucagon and secretin. Okay, so we have to name that peptide hormone which acts on hepatocytes, that is liver cells, adipocytes, that is cells of fatty tissues, and enhances the cellular glucose uptake and utilization. Okay, so if these are the cells and here we have blood, so this hormone is enhancing the cellular glucose uptake. That means the glucose will be taken up by the cells. Okay, the glucose present in the blood will be taken up by the cell. That means the amount of glucose in blood will decrease. Okay, so glucose level in blood will decrease. And which hormone causes decrease in blood glucose level? Insulin. Okay, so the correct answer will be option number B. The glucagon enhances the blood glucose level, okay, or it increases the blood glucose level. Okay, so the correct option is option number B. Question number 75 is from the chapter Locomotion and Movement, and the question is osteoporosis, an age-related disease of skeletal system, may occur due to option A, accumulation of uric acid leading to inflammation of joints. This is about the disease known as gout, not osteoporosis. Okay, so in case of gout, there is inflammation of joints that is due to the accumulation of uric acid crystals. Okay, then the second one is immune disorder affecting neuromuscular junction leading to fatigue. So this is about another disease known as myasthenia gravis. Okay, myasthenia gravis, which is an autoimmune disorder. Okay, and this leads to fatigue, weakening, and paralysis of skeletal muscles. Then the third one is high concentration of calcium ion and sodium ion. This is also incorrect. And the last one is decreased level of estrogen. Yes, this is correct. Okay, so with increase in age, the level of estrogen decrease and it causes osteoporosis, which is characterized by the lower bone mass or decreased bone mass and increased chances of fractures. Okay, increased chances of fractures. So option D will be the correct answer. Question number 76 is from the chapter Body Fluids and Circulation, and the question is serum differs from blood in option A lacking antibodies, option B lacking globulins, option C lacking albumins, and option D lacking clotting factors, and the correct answer is option D lacking clotting factors. Okay, so serum along with clotting factors make up plasma, blood plasma. Okay, so serum along with the clotting factors makes blood plasma. Okay, so serum will be equal to blood plasma minus clotting factors. Okay, that means if we remove the clotting factors from the blood plasma, we'll get the serum. Okay, so this is the difference, that the serum lacks clotting factor. So option D is the correct answer. Question number 77 is from the chapter Breathing and Exchange of Gases, and the question is lungs do not collapse between breaths and some air always remains in the lungs which can never be expelled because option A, pressure in the lungs is higher than the atmospheric pressure, option B, there is a negative pressure in the lungs, option C, there is a negative intrapleural pressure pulling at the lung walls, and option D, there is a positive intrapleural pressure. Okay, so we know that the lungs are double membraned organs. That means these are lined by two membranes which are known as pleural membranes, right? Pleural membranes. And there exists a pressure between these two membranes that is known as intrapleural pressure. Intrapleural pressure. And this is about 756 mm of Hg. Whereas the atmospheric pressure is 760 mm of Hg. So we can say that this intrapleural pressure is less than the atmospheric pressure, and by what amount? By 4 mm of Hg. Okay, so intrapleural pressure is considered as minus 4 mm of Hg because we are comparing it with the atmospheric pressure, right? So it is considered as negative pressure. And due to this negative pressure, the air will move towards the lungs. Why? Because air always moves from higher pressure to lower pressure. So movement of air will be from the atmosphere because atmosphere has a higher pressure towards the lungs. Okay, and this amount of air will always remain in the lungs whether the process is inspiration or expiration. Okay, therefore the correct answer will be option number C. There is a negative intrapleural pressure pulling at the lung walls. Okay, Question number 78 is from the chapter Chemical Coordination and Integration, and the question is, the posterior pituitary gland is not a true endocrine gland because option A, it secretes enzymes, option B, it is provided with a duct, option C, it only stores and releases hormones, and option D, it is under the regulation of hypothalamus. And the correct answer is option C, that it only stores and releases hormones. Okay, we know that true endocrine gland synthesize hormones, but posterior pituitary does not synthesize hormones. Okay, so posterior pituitary, that is also known as neurohypophysis. Neurohypophysis, it releases two hormones, namely oxytocin and ADH. Okay, but these two hormones are not synthesized by the posterior pituitary. These two hormones are synthesized by the hypothalamus. Okay, so the synthesis takes place in hypothalamus. That means posterior pituitary does not synthesize any hormones, therefore it is not a true endocrine gland. It only stores and releases hormones. Okay, so the correct answer will be option number C. Okay. Question number 79 is from the chapter Excretory Products and Their Elimination, and the question is, the part of nephron involved in active reabsorption of sodium is. Options are descending limb of Henle's loop, distal convoluted tubule, proximal convoluted tubule, and Bowman's capsule. Okay, so what is a nephron? It is the functional unit of kidney. Okay, functional unit of kidney is known as nephron. It has different parts such as Bowman's capsule, DCT or distal convoluted tubule, PCT or proximal convoluted tubule, and Henle's loop. Okay. So here the question is about the active reabsorption of sodium. So we know that active reabsorption of sodium takes place in two tubules of the nephron. First one is proximal convoluted tubule and the second one is distal convoluted tubule. Okay. So DCT as well as PCT. Now, for answer we have to choose any one. So it will be the third one, that is proximal convoluted tubule, because here maximum reabsorption of sodium takes place around 67%. Okay, and then the reabsorbed sodium is sent to the peritubular capillary. Okay, then it is sent to peritubular capillary. Now, why not DCT? Because the reabsorption in DCT is only conditional. Okay, it is not regular, it is only conditional. That means it happens only when required by the body. Okay, also it is under hormonal influence or under hormonal regulation. Therefore the correct answer will be option number C, PCT or proximal convoluted tubule. Question number 80 is from the chapter Reproductive Health, and the question is, which of the following is hormone releasing IUDs? Options are copper 7, LNG 20, multi-load 375 and Lipis loop. Okay, so what are IUDs? These are a type of contraceptive devices and it stands for intrauterine devices. Okay, intrauterine devices. There are mainly three types of IUDs. First one is non-medicated IUDs. Then the second type is copper releasing IUDs. And the third type is hormone releasing IUDs. Okay, hormone releasing IUDs. So in the options we have copper 7. This is an example of copper releasing IUDs. LNG 20 is an example of hormone releasing. Okay, hormone releasing. So this will be the correct answer because the question has been asked about the hormone releasing IUDs. Multi-load 375 is also a copper releasing, and Lipis loop is an example of non-medicated. Okay, non-medicated. So the correct answer will be option number B. Question number 81 is from the chapter Reproductive Health, and the question is, which of the following is incorrect regarding vasectomy? So what is vasectomy? It is a surgical method of contraception. Okay, vasectomy is surgical method of contraception in males. Okay, in female it will be tubectomy, but in males what happens? The vas deferens is cut. Vas deferens or vas deferentia is cut and tied. Okay, like this. So this is the vas deferens. Then after vasectomy it will be cut here and then tied on both sides. Okay, like this, right? So that the sperms do not pass through the vas deferens. Okay, it is done to stop the passage of sperms. Stop the passage of sperms through the vas deferens. Okay, through vas deferens. Now we know that the sperms are formed in the testis and then they move to the epididymis. Okay, epididymis, and after that they move to the vas deferens or vas deferentia. So we know that in vasectomy, this vas deferens is cut. That means before vas deferens, the sperms should be available. Okay, so sperms will be formed in testis as usual. They will pass through epididymis also, but they will not be able to pass through vas deferens. Okay, now let's go through the options here. The first option is irreversible sterility. Yes, so this process or this procedure is intended to be permanent. Okay, therefore this is correct. Then the second statement says no sperms occurs in seminal fluid. This is also correct. Okay, so after this procedure also seminal fluid will be formed and it can be ejaculated as well, but it will not contain the sperm with it because the sperm has been stopped in the vas deferens only. Okay, then the third one is no sperm occurs in epididymis. This is incorrect because we have seen that before vas deferens sperms will occur. Till epididymis the sperms will occur. Okay, so this is incorrect. Then the last one is vas deferentia is cut and tied. Yes, this is also correct. So the only incorrect one is option number C. So it will be the correct answer. Question number 82 is again from the chapter Reproductive Health, and the question is, embryo with more than 16 blastomeres formed due to in vitro fertilization is transferred into. Options are cervix, uterus, fallopian tube and fimbriae. So we know that in case of in vitro fertilization, the sperms are made to fuse with the ovum outside the female body. That is, fertilization takes place outside the body. Okay, so fertilization outside the female body. That means zygote will also be formed outside the body. Zygote formation takes place outside the female body. Okay, now this has to be transferred into the body. Okay, transfer into the body. So this transfer can take place in two different ways. In the first case, when there is early embryo up to 8 blastomeres, then it can be transferred into the fallopian tube. Okay, fallopian tube, and from there it will travel to the uterus. Okay, and this process is known as zygote intrafallopian transfer or ZIFT in short. Okay, whereas in the second case, when there is more than 8 blastomeres. Okay, so here 16 blastomeres are mentioned. So for 16 blastomeres, it can be transferred directly into the uterus. Okay, into the uterus, and this is known as intrauterine transfer or IUT in short. Okay, so for 16 blastomeres it has to be transferred inside the uterus. Okay, so the correct answer will be option number B. Question number 83 is from the chapter Human Reproduction, and the question is, which of the following depicts the correct pathway of transport of sperms? Okay, so we know that the sperms are produced in the seminiferous tubules of the testis. Okay, seminiferous tubules. So here the sperms are formed inside the testis. After that, these are transferred to the network of ducts known as reti testis. Reti testis. After this, they transfer into the duct known as vasa efferentia. So efferentia, and this is also known as efferent ductules. Efferent ductules. After that, they pass through highly coiled duct known as epididymis. Okay. And then the next duct is vasa differentia. And this vasa differentia will further direct the sperms towards the ejaculative duct. Okay, so let's see. In the first option, we have efferent ductules first and then reti testis, which is not true because first there will be reti testis and then efferent tubules. Okay, so this is incorrect. Then in the second one, we have reti testis, yes. Then efferent ductules, then epididymis, and then vas deferens. Yes, this is the correct sequence. So this will be the correct answer. Okay. Question number 84 is from the chapter Human Reproduction, and here we have to match column 1 with column 2. Okay, so the first one mentioned in column 1 is mons pubis. Mons pubis. So what is mons pubis? It is a part of female external genitalia and it is basically a mass of fatty tissue, that is covered by skin and pubic hair. Okay, covered by skin and pubic hair. So A will be matched with number 3 here. Okay, match for A will be number 3. Then the second one is antrum. Antrum. Now what is antrum? It is the portion of Graafian follicle. Portion of Graafian follicle that is filled with follicular fluid. Filled with follicular fluid. So it will be matched with number 4 in column 2, that is Graafian follicle. Okay, match for B will be number 4. Then the third one is trophoectoderm. Trophoectoderm. It is found on the outer edge of the blastocyst. On the outer edge. Okay, these are the cells. Okay, sequence of cells which are found on the outer edge of the blastocyst. And these are the first to differentiate. In order to form a fertilized egg and further help in embryo development. Okay, so it will be matched with embryo formation. Okay, because this is involved in the embryo formation. So the match for C will be number 1. And the last one is nebenkern. Nebenkern. So what is nebenkern? It is derived from the mitochondria and it is a two-stranded helical structure. Two-stranded helical structure in the proximal end of the tail region of the sperm. Okay, tail region of sperm. So basically this is part of sperm. So it will be matched with number 2, that is sperm. Okay, so the match for D will be number 2. Now let's see which will be the correct answer. A3, B4, C1 and D2. So option C will be the correct answer. Question number 85 is again from the chapter Human Reproduction, and the question is several hormones like HCG, that is human chorionic gonadotrophin, HPL, that is human placental lactogen, estrogen and progesterone are produced by. Options are pituitary, ovary, placenta and fallopian tube. Okay, so we know that pituitary gland produces several hormones, but these 4 are not among them, so this cannot be the correct answer. Ovary produces estrogen and progesterone. Estrogen and progesterone, but not HCG or HPL, so this is also incorrect one. Then the third one is placenta. Yes, placenta will be the correct answer because all these four hormones are produced by the placenta. Also in the later stage of pregnancy, the placenta releases or produces relaxin along with these four hormones. Okay, and ovary also releases relaxin at the later stage of pregnancy. Okay, and the last one, that is fallopian tube. So fallopian tube cannot be the correct answer because it does not produce any hormone. Does not produce any hormone. So the only correct answer is option number C, placenta. Question number 86 is from the chapter Principles of Inheritance and Variation. And the question is, if a color-blind man marries a woman who is homozygous for normal color vision, then the probability of their son being color-blind is. Options are 1, 0, 0.5 and 0.75. Okay. So, first of all, we should know that color blindness is an X-linked trait. Okay. X-linked trait. So this is the most important point here. X-linked trait means it will be found only on the X chromosome, only on X chromosome. Okay, let's write XC for the chromosome having this trait. Okay, now we have a man here and the genotype of man we know it is XY, but since this man is color blind, therefore the X chromosome here will contain this trait. Okay, trait for blindness. So let's write XC for this. Okay, so the genotype of this man will be XCY, and what will be the genotype of the woman? It will be normal XX. Okay, now let's draw the Punnett square here to see what will be the genotype of the progeny. Okay, so here we have XC for male and Y for male, and this side the genotype of the female, that is X and X. Okay, so let's see what will be the genotype of the progeny. Here we get XXC, here we get XY, then again XXC, and then XY. Okay, now these two XY are males or sons, whereas these two XX are females or the daughters. So we can see that this trait for blindness is present in daughters but not in sons. Okay, but the question is asked that the probability of their son being colorblind is, so it will be 0 because the males don't have any trait here. Okay, so option B will be the correct answer. Question number 87 is from the chapter Evolution, and the question is, genetic drift operates in option A, slow reproductive population, option B, small isolated population, option C, large isolated population, and option D, non-reproductive population. Okay, so what is the definition of genetic drift? Genetic drift is the random change, let's write it down here, it is the random change in the allele frequency of a small population. Okay, small population or small and isolated population. Therefore the correct answer will be option number D. So this is a direct question, definition based question. Okay, option B will be the correct answer. Question number 88 is again from the chapter Evolution, and the question is, in Hardy-Weinberg equation, the frequency of heterozygous individual is represented by, options are Q square, P square, 2PQ and PQ. Okay, so we know that according to the Hardy-Weinberg principle, alleles and genotype frequencies in a population will remain constant from generation to generation in the absence of any other evolutionary influences. Okay, and he gave the equation known as Hardy-Weinberg equation, that is P plus Q whole square, where P plus Q is equal to 1. Okay, now if you open the bracket, P plus Q whole square, then we'll get P square plus Q square plus 2PQ, right? And we know that P plus Q is equal to 1. So P plus Q whole square will also be 1. So the value of P square plus Q square plus 2PQ will be equal to 1. Okay, so this is the equation. Now, we know that for the expression of any character, genes are required, and each gene has two alleles, okay? And these two alleles are present on the homologous chromosome. So, suppose these are two homologous chromosomes. So, the alleles are present on the same locus on these chromosomes in alternate form, okay? Now, three conditions are possible. So, let's draw three different diagrams here. In the first condition, both the alleles can be dominant alleles. So here P represent the dominant allele. So okay, so in the first condition it can be PP. In the second condition, both the alleles can be recessive. So Q represents the recessive allele. Okay, so QQ for the recessive. So the first condition is known as homozygous dominant condition, and the second one is known as homozygous recessive condition, right? And in the third condition, one can be dominant and the other can be recessive. So it can be P and Q. Okay, and this is known as heterozygous condition. Heterozygous condition, because both the alleles are different. So here the question is asked about the heterozygous individual. Okay, that means about this third equation, third condition. Okay, so the answer will be 2PQ, because here according to this equation we have 2PQ for the heterozygous individual. So option C will be the correct answer. Now question number 89 is also from evolution, and the question is the chronological order of human evolution from early to recent is option A Australopithecus, then Homo habilis, then Ramapithecus and Homo erectus. Okay. Option B Australopithecus, then Ramapithecus, then Homo habilis and Homo erectus. And then the third one is Ramapithecus, then Australopithecus, then Homo habilis and then Homo erectus. And the last one is Ramapithecus, Homo habilis, Australopithecus and Homo erectus. Okay. So the correct answer out of them will be option number C. First one will be Ramapithecus. Okay. Let's write it down here. Ramapithecus. These were also known as ancient apes. Okay. Ancient apes, and these were found 12.2 million years ago. Then the second one is Australopithecus. Australopithecus. These were found in Africa and they were found around 4.2 million years ago. Then the next one is Homo habilis. Homo habilis. These were known as handy man. Okay, handy man. These were found in the Pleistocene era. Pleistocene era, and around 2.3 million years ago. And the last one is Homo erectus. These were also found in the Pleistocene era, and the timeline was around 1.5 million years ago. Okay, so the correct sequence will be first Ramapithecus, then Australopithecus, then Homo habilis, and at last we have Homo erectus. So option C will be the correct answer. And now the last question is again from the chapter Evolution, and the question is which of the following is the correct sequence of events in the origin of life? First one is formation of protobions, then synthesis of organic monomers, then synthesis of organic polymers, and last is the formation of DNA based genetic system. Okay, so first one is protobions. Protobions are the first formed cells. First formed cells. But for the formation of cells, organic compounds or organic molecules are required. Okay, so before one, there will be the formation of organic compounds. Now organic polymers and monomers both are there. So for the formation of polymers we need monomers, right? Because these are formed from the monomers only. So before 3, 2 will come. Okay, so first 2, that means the formation of organic monomers, then 3, that is formation of organic polymers, and then these will aggregate to form the cells. So first one, okay, and at last the genetic system would be formed. So fourth one will be the last. So this is the sequence 2, 3, 1 and 4. Therefore the correct one will be option number D. Okay, this is the correct answer. So here we complete all the biology questions from NEET 2016 phase 2 paper. Okay, and we'll be back with another session very soon. Till then, keep learning, keep scoring high. For a better understanding, I would suggest you to practice the same questions from my website www.examsnet.com, and for more information you can download our app from the link given in the description. Thank you so much.